Hi everyone, welcome to my channel Pure Biology, and today's topic is receptors, and this is obviously the topic from cell signaling. And before going to this topic, I want to say that already I have uploaded the two videos related to cell signaling, so you just check out that, and after this that you just check out this one, okay. And obviously I request you that please you subscribe and share my channel and hit the bell icon for getting the notifications for my all videos, okay. So let's continue. This is the receptors, okay. So in my previous video I already described about the signals that are very important for cell signaling process, and here this is the cell signaling receptors, okay. And these cell signaling receptors are what? The cellular response to a particular extracellular signal molecules that depends on its binding to a specific receptor located on the surface of a target cell or in its nucleus or cytosol. And receptors are chemically protein or glycoprotein molecules. These are called the ligand. So what is saying that if the receptor is not present on the target cell, that signals cannot bind with the receptor of the target cell, and so signaling process or signal transduction is not occurring in the target cell. So receptor is very important for continuing the signaling process. So generally this the response that is occurring due to the signal when the signal just bound to the receptors, specific receptors, okay. And the specific receptor that remain either on the surface of the cell or in the nucleus or in the cytosol. Clear? So if these are present in the surface, or these are known as the cell surface receptors, and if these are present in the nucleus and cytosol, so these are called the nucleus or cytosolic receptors. So how this and the receptors, these are the actually structurally or chemically these are the protein, okay, or maybe glycoprotein. So these are called also ligand. And here the binding of a ligand, when ligand bind to any signal, then this causes a conformational change in the receptor, and that initiates a sequence of the reactions and leading to the specific cellular response based on the location, and the receptors are classified, okay. This is another part. So just up to this, what is saying that when any signal that is bound to the receptor of target cell, then a conformational change is occurring, okay. And why this conformational change is occurring? Because when the specific signal bind to the specific receptor, if the conformational change is not occurring, then another signal may be bound to the receptor, okay. And in that situation the signaling transduction or the signaling process may be hampered. So that's why when the signal, any signal that bound to the already bound to the receptors of the target cell, there is a conformational change is occurring, okay. And this conformational change, due to this conformational change, the specific cellular response is seen, okay. Specific cellular response is seen. And the based on the location, the receptors are two types, generally intracellular receptor and the cell surface receptors. So first we have to learn about the intracellular receptor, then we will learn about the cell surface receptor, okay. So intracellular, so intracellular receptor, these are located in the cytosol or the nucleus, And this include the receptors for steroid hormones. Okay, steroid hormones. So steroid hormones are very important for these receptors, and these receptors belong to the nuclear receptor or short form NRS. This is called the NRS superfamily. That means nuclear receptors superfamily. These are the family of the highly conserved transcription factors and that regulate the transcription in response. Okay, these are the actually related to the transcription factors, and obviously these regulate the transcription process. Obviously, when transcription process is arised by this, when any small lipophilic signal molecules may be bound to the NRS or the nuclear receptors, and these receptors consist of generally N-terminal activation domain, a central DNA binding domain, and C-terminal ligand binding domain. Okay, so these consist of three domains. One is the N-terminal domain, that is called the AB region, central DNA binding domain, that is called the DBD, and the C-terminal ligand binding domain, that is called the LBD. Clear? So here this N-terminal domain that contains AF1. This AF1 is a full form is that activation function 1 sequence, and that functions as a ligand that independent transcriptional activators. Okay, so actually what's the function of this? This functions as a ligand independent transcriptional activator. Okay, acts as a transcriptional activator. And next one is the DBD, that is comprised of two zinc binding motifs, and that each coordinate, that means one zinc binding motif that coordinate a zinc ion through 4 positions cysteines. Okay, it coordinate one, but that means two among the two zinc motif, one coordinate with the zinc ion, one coordinate with the zinc ion, one zinc ion, through 4 cysteines. Okay, through 4 positions cysteine. And the third one is that LBD. This is comprised of the AF2, like N-terminal domain. This domain composed of AF2. This is called the activation function 2 sequence, and that is necessary for what? Recruiting the transcriptional co-activators. Okay, this help to recruit the transcriptional co-activators. So what are the functions of the 3 domains? First one that contains AF1, and its functions are it acts as the transcriptional activator. Okay, and the second one is that consists of the 2 zinc motif, and among 2 zinc motif, one coordinate with one zinc ion if there are 4 cysteine present there. Okay, so it is possible if 4 cysteines present there, then one zinc motif that coordinate with one zinc ion. And the last one LBD, that consisting of AF2, which is necessary for what? That helps in recruiting the transcriptional co-activators. Okay, these are the actual functions of the domains. This also contributes to homodimerization and heterodimerization of nuclear receptors. Okay, nuclear receptors. So here homodimerization process and heterodimerization process, and that totally done by the nuclear receptors. And these nuclear receptors act as the regulatory transcription factor. So it is already said there, and with the regulatory DNA sequence, and this sequence are called the response element. Okay, and these remain as monomers, homodimers or heterodimers. Okay, so one second. So what is that? This response element is what? Response element, when the NCS receptor or the nuclear receptors act as the regulatory transcription factors and bind with the regulatory DNA sequence, then it is called the response element. Okay, and generally they can bind with the response element as monomer, homodimer, or the heterodimers. Okay, so this is the actual topic of intracellular receptors. This is a long topic. I think there is a space is very short, so I have to write the next part about this intracellular receptor. Okay, so continue with my channel. So here the thing is that one example is taken here, that when the steroid hormones, these are generally located on the cytosol and it remains in inactive form. That means when it is located on the cytosol, it remains in inactive form. Okay, and after making a complex with the heat shock protein, when it make a complex with the HSP protein or ligand binding with it, and already ligand also binding with it, in that situation, the cytosol, in the dissociation of the HSPs and translocation from cytosol into nucleus. That is what is happening. First of all, three are actually binded with each other. One is the heat shock protein, another is the ligand, and another is the receptor with ligand. Okay, so when the three make a complex, then the cytosol, they first remain in the cytosol, but when the dissociation of the HSPs, the translocation occurring from cytosol to into the nucleus. Okay, it's understood that first of all, suppose this is the steroid hormone, it binds to the receptor that remain on the cytosol. Okay, but in that situation, ligand also remain with the receptor, but they all totally in inactive form. When heat shock protein is added, they are activated, and after that, what is happening? Just after dissociation of the HSPs or the heat shock protein, the hormone or the steroid hormone that enters from the cytosol to the nucleus. Okay, once this ligand receptor complex is in nucleus, after just entering into the nucleus, its DNA binding domain to receptor binds to the specific hormone receptor element of the DNA. Okay, and that means that when a heat shock protein is already there with the receptor and the steroid hormones, they remain in the cytosol. Just after releasing the heat shock protein, only the hormone with the ligand receptor enter into the nucleus, and when it enter into the nucleus, they bind to the specific DNA, specific element of the DNA. Okay, so this is actually happening in case of the steroid receptor. Okay, and here the steroid receptor, this acts like the homodimers. Clear? And here the identity of the response element resides in the three features. Okay, so sequence of the base pairs in the half site, number of base pairs between the two half sites, and the relative orientation of the two half sites. So these features first have to identify by the response element. Okay, or you can say that we identify the response element with these three features. With these three features, we can easily identify of a response element, because response elements have the three features. The sequence of the base pairs that in half sites, and number of base pairs between two half sites, okay, between two half sites. And third one is that relative orientation of two half sides. Okay, relative orientation of two half sides. So each receptor protein dimer that binds to DNA has to recognize the sequence, spacing and orientation of the half sides within their response element. So receptor proteins must have to, mainly the receptor protein, they has to identify what that the sequences, spacing and the orientation of the half side of what? Of the response element. Okay, of the response element. Without recognition of these three features, the binding is not possible. Okay, without recognizing three features, the binding is not possible. So, receptor protein dimers that binds to DNA has to recognize these three features of the response element. In case of, this is occurring in the steroid hormone, but in case of thyroid hormone, what is happening? This is not a steroid hormone. In that situation, what is happening? Here, thyroid hormone receptor and the RXR means retinoid X receptor, they bind to DNA half sides of the hormone response element. Clear? And they form heterodimers. Here the steroid hormone, there we can see that they form homodimers. Okay, but here they form heterodimers. Why heterodimers? Because here two receptors are there. One is the thyroid hormone receptor and another is the retinoid hormone, retinoid X receptors. Okay, both two bind to the DNA half side of the hormone response element. Here only one is important, but here two are important for the binding with the response element. Clear? And they form, that's why they form the heterodimer. In absence of thyroid hormone ligand, TR means thyroid receptor that recruits the co-repressor protein, one co-repressor protein. Okay, co-repressor protein. Sorry. Co-repressor protein. To inhibit the transcription. Okay, to inhibit the transcription. So what is happening here, that TR, in absence of thyroid hormone ligand, if ligand is not there, then what is happening, this thyroid receptor, this recruits co-repressor protein to inhibit the transcription, and TR and RXR that bound to DNA but hormone response element is inhibited. Okay, so why the hormone response is inhibited? Actually due to the recruitment of the co-repressor protein, the hormone response element is inhibited. Okay, that means here the ligand is absent. That's why TR already recruits the co-repressor protein to inhibit the transcription. So TR and RX are already bound to this DNA, but here the hormone response element is inhibited. So no transcription is occurring. But when the ligand is present, what is happening? We will see in the next part. So while the ligand is bind with the TR or TR binds to the ligand, in that situation what is happening? The co-repressor, that proteins are removed and degraded by the proteasomes. Okay, and while co-activators are recruited for continuing the transcription process. Okay, so this is the activation of the receptors and signals in steroid hormones and in case of non-steroid hormone. These are the two examples explained here. So I will try to draw a rough sketch of the two hormones. The one is the steroid for steroid hormone and non-steroid hormone in my next part. Next part, not in this part, just rubbing this portion. So here, this is the rough structure. Here, this is the steroid hormone just enter with the receptor, and HSP already bound with that, is already bound with that. So first of all, HSP is released, and then this steroid hormone receptor enter and bound to with the hormone response element to continue the transduction. And here you can see that ligand with receptors. One second. Okay, these are the thyroid hormones enter and that bound to the receptors and ligand and receptors. And here the heterodimer is formed because with the thyroid hormone receptor there is another hormone receptor is there, that is the RXR. So that's why this is the heterodimer, and here only one receptor is there, that's it is the homodimer. Okay, so thyroid hormone and receptor that bound with the response element, and that's why the transduction is occurring. Okay, transduction is occurring. So this is the actual happening, okay, or this is occurring in the cell. The actual role of the receptor is diagrammatically very briefly or very roughly I have drawn here how this is occurring. Okay, so you just continue with my channel for getting the another video or another topic in the next video. So thank you.